Hello everyone, this is Dr. Divya. Topic for today's discussion is biosynthesis of heme. Heme is produced by the combination of iron with a porphyrin ring. Chlorophyll 2, the photosynthetic green pigment in plant, is magnesium porphyrin complex. Heme is present in hemoglobin, myoglobin, cytochromes in ATC, peroxidase, catalase, nitric oxide synthase. Hemoglobin is a conjugated protein having heme as a prosthetic group and the protein, the globulin. Porphyrin contains four pyrrole ring A, B, C, and D joined through methyl bridges. Porphyrin contains side chains attached to each of the four pyrrole rings. In type 1 porphyrin, the side chains are arranged symmetrically, that is for uroporphyrin 1. Acetate alternates with propionate around the tetrapyrrole ring. An acetate and propionate are reversed in ring D of uroporphyrin 3 when compared with uroporphyrin 1. Only type 3 porphyrins are physiologically important in human. This is the structure of heme. And side chains present in the heme. Different porphyrin vary in the nature of the side chains attached to each of the four pyrrole ring. First one is uroporphyrin, contains acetate and propionate side as a side chain. Coproporphyrin contains methyl and propionate groups. Protoporphyrin 9, which is heme B, the most common heme, contain vinyl, methyl, and propionate groups. This is the structure of heme. Side chain distribution. The side chain of porphyrin can be ordered around the tetrapyrrole nucleus in four different ways, designated by Roman numerals 1 to 4. Only type 3 porphyrin, which contain an asymmetric substitution on ring D, are physiologically important in humans. Porphyrinogen. These porphyrin precursor, for example uroporphyrinogen, exist in a chemically reduced colorless form and serve as intermediates between porphobilinogen and the oxidized colored protoporphyrin in heme biosynthesis. Biosynthesis of heme. Heme can be synthesized by almost all the tissues in the body, most active in bone marrow, that is 85%, and liver. Heme is not synthesized in the matured erythrocytes. The pathway is partially cytoplasmic and partially mitochondrial. Step 1, synthesis of ALA. The synthesis starts with the condensation of succinyl-CoA and glycine in the presence of pyridoxal phosphate to form delta aminolevulinic acid. Hence, anemia may be manifested in pyridoxal deficiency. The enzyme ALA synthase is located in the mitochondria and is the rate-limiting enzyme of the pathway. Step 2, formation of porphobilinogen. Next few reaction occurs in the cytoplasm. Two molecules of ALA are condensed to form porphobilinogen, that is PBG. The condensation involves removal of two molecules of water and the enzyme catalyzes this reaction called ALA dehydratase. Porphobilinogen is a monopyrrole and the enzyme contains zinc and is inhibited by lead. Step 3, formation of uroporphobilinogen. Condensation of four molecules of porphobilinogen leads to the formation of first porphyrin of the pathway, namely uroporphyrinogen. Condensation of porphobilinogen produces a linear tetrapyrrole hydroxymethylbilane. The enzyme for this reaction is catalyzed called hydroxymethylbilane synthase, and this hydroxymethylbilane molecule will cyclize and isomerize to form uroporphyrinogen 3. Hydroxymethylbilane is converted to uroporphyrinogen 3 by the enzyme called uroporphyrinogen 3 synthase, and during this deamination reaction, 4 molecules of ammonia are removed. Step 4, synthesis of coproporphyrinogen. The uroporphyrinogen 3 is next converted into coproporphyrinogen 3 by decarboxylation. 4 molecules of carbon dioxide are eliminated and the enzyme catalyzes this reaction called uroporphyrinogen 3 decarboxylase. The acetate group are decarboxylated into methyl group. Step 5, synthesis of protoporphyrinogen. And further metabolism takes place in mitochondria. Coproporphyrinogen is oxidized to form protoporphyrinogen 3 by coproporphyrinogen oxidase enzyme. This enzyme specifically acts only on type 3 series and not on type 1 series. Two propionic acid side chains are oxidatively decarboxylated to form vinyl group, and this reaction requires molecular oxygen. Step 6, generation of protoporphyrin. The protoporphyrinogen 3 is oxidized by the enzyme called protoporphyrinogen oxidase to form protoporphyrin 3 in the mitochondria. The oxidation requires molecular oxygen, and the methylene bridges are oxidized to form methine bridges, and colored porphyrins are formed. Step 7, generation of heme. The last step in the formation of heme is the attachment of ferrous ion to the protoporphyrin. The enzyme is ferrochelatase or heme synthase, which is located in mitochondria. Iron atom is coordinately linked with 5 nitrogen atoms, that is 4 nitrogen of pyrrole ring of protoporphyrin and first nitrogen atom of a histidine residue of globin. The remaining valency of iron atom is satisfied with water or oxygen atom. When the ferrous ion in heme gets oxidized to ferric form, hematin is formed, which loses the property of carrying the oxygen. Heme is red in color, but hematin is dark brown in color. This is the overall reaction of biosynthesis of heme. Moving on to the regulation of heme synthesis. ALA synthase is a key rate limiting enzyme. Heme, lead poisoning, and steroids inhibit its activity. Excess heme in BM is converted to hematin to oxidation of Fe2 plus to Fe3 plus. ALA synthase is also allosterically inhibited by hematin. ALA synthase is activated by hypoxia due to increase in erythropoietin. ALA synthase is also activated by availability of intracellular iron. Isonicotinic acid hydroside that decreases the availability of pyridoxal phosphate may also affect heme synthesis. And drugs like barbiturates induce heme synthesis. Barbiturates require the heme containing cytochrome P450 for their metabolism. Out of the total heme synthesized, two-thirds are used for cytochrome P450 production. Porphyrias, a group of disease associated with abnormalities in the biosynthesis of heme, characterized by accumulation and excretion of porphyrin or porphyrin precursors. Most inherited porphyrias are autosomal dominant except one. Thank you.